Good afternoon. My name is Beata Lubkowska, and I work at the Department of the Molecular Biotechnology, and I'm waiting all day for this presentation, and it's just difficult. Right. My first okay, let me start about the basic. Based on the very recent research in Poland at the University of Gdańsk on thermophilic TP84 bacteriophage infecting Geobacillus styrothermophilis bacteria, showing the uniqueness of the sequence 47.7 KBP genome and the lack of its significant homology to the genomes of other bacteriophages, as well as preliminary proteomic studies. A new genus TP84 virus was created and defined, established specifically for TP84 by the International Committee on Taxonomy of Virus, ICTV, in the Syphoviridae family. This virus is a bio-nano platform in one of our projects, as shown on the left side on the figure. The activity of TP84 glycosylase depolymerase enzyme is an important aspect of the biology of host infection by this bacteriophage. This is because Geobacillus styrothermophilis produces thick envelopes that greatly exceed the vegetative cell volume by approximately 50 times, and their thickness exceed the length of the bacteriophage tens of times. Therefore, TP84 bacteriophage apparently has an effective mechanism allowing for penetration of these protective shells. As you can see on the screen, exactly, bacteriophage TP84 is a primary source of depolymerase glycosylase about code TP8426, and it is the biggest protein TP84, which has a molecular mass 112.2 kilodalton. We obtained the glycosylase depolymerase by several procedures and evaluated the yield of the native protein obtained by densitometric analysis. Quantitative measurements of DNA and proteins in bands made using a program unscanned. The concentration of glycosylase in the concentrated preparation was determined by scanning of Kumasi brilliant blue stained SDS page gels to be 2 milligram per milliliter. And also on figure 4, the native the enzyme samples on the right indicate that the glycosylase enzyme is a multimer. The enzyme samples on the left indicate that the multimer has dissociated and migrated in the gel as a smaller protein species. So we obtained the glycosylase depolymerase expression clones, which enable their genetic modification with the His6 tag for metal affinity chromatography purpose, yielding ultra-pure enzyme. Genetic modification of the clones were introduced by site-specific mutagenesis using a mutagenic primers PCR method, gene cloning and expression recombination with restriction endonucleases and DNA modification enzymes. Also monitoring gene expression by quantitative SDS page. Since the codon usage in the TP84 genome is well tolerated by Escherichia coli bacterium, use recombinant cloning and genetic expression vehicle, there is no need to optimize the TP84-26 gene codons. Designer PCR primers on figure 6, amplifying restriction endonuclease, and also the coding open ORF is in such a way that DNA sequence recognized by BSA and cell restriction endonuclease are introduced at the 5 and 3 ends of the gene, which will allow for directional cloning of the TP84-26 ORF in perfect fusion with this vector, with the vector and start codon. The primers were designed so that no additional codons were introduced. If you see on figure 7, what there is shown exactly, the TP84-26 gene PCR amplification optimization was conducted by testing various annealing temperatures, those yielding clean reaction products comparison the designer gen. The PCR product was digested with BSL and cell gel purified, ligated with the modified PET21D plus vector containing his 6 tag following the vector start codon at the N terminus. But 21 D6, D6. The PCR cloning was devised in such a way that the used restriction enzymes did not introduce additional amino acids residues to the cloned ORF. In other words, precisely the native glycosylase depolymerase polypeptide sequence was cloned into the modified PET21D plus vector, forming the N-terminal fusion with his 6 tag. The cloning has resulted in a large number of clones, and the culture continues to steadily grow after induction, indicating that the TP84-26 gene is not toxic to the recombination recombinant of Escherichia coli host. The selected clone shown on figure 9 exhibition, the highest expression was used for the protein, upscaled, biosynthesis, and purification. The his 6 tag TP84-26 fusion variant was expressed and isolated by a combination of selective precipitation of nucleic acids with polyethylamine and ammonium sulfate. Also, we use affinity chromatography on blue sepharose, anion exchange chromatography on NGC rescue, affinity chromatography on heparin sepharose, and repeated anion exchange chromatography on NGC rescue, yielding nearly homogeneous protein. Interestingly, even though the recombinant His6 glycosylase depolymerase was constructed for the use of originally intended metal affinity chromatography with immobilized nickel anions, the protein did not bind to this chromatography media, which suggests that the His6 tag is buried within the protein structure. This result collaborates with the recombinant His6 glycosylase depolymerase behavior on Western blots using antisera developed against entire TP84 bacteriophage particles. The intact His6 glycosylase depolymerase react very poorly with the antibodies. In spite of that, on SDS page, the recombinant enzyme band contains large amount of the protein. To the contrary, it's apparently proteolytic fragment, but I'm not show it here. But this fragment was a little stronger than our properly answer. As the TP84 plaques halo size increases with incubation time, it is apparently caused by glycosylase depolymerase diffusion faster through agar than the TP84 bacteriophage particles. This shows that glycosylase depolymerase is also present in not bound to TP84 particles form. We hypothesize that the enzyme is associated as a structural protein in the region of tile fibers and base plates, allowing for direct perforation of the Geobacillus styrothermophilus thick capsule. Further, we confirm that the enzyme degrades the capsule, but not the bacteria cell wall. We perform a series of studies on thermostable TP84 bacteriophage glycosylase depolymerase activity as the function of the bacteriophage plaques grow incubation time. The growth range of TP84 bacteriophage ranges from 43 exactly to 76 Celsius degrees and is closely related to the growth of the host Geobacillus styrothermophilus. The morphology of the phages of the bacteriophages plaques varies with changes in temperature and humidity, as can be seen in the figure 14. Small, large, and exceptionally large multi-layer plaques with or without halo, and also iridescence halo are observed. TP84 depolymerase glycosylase is either expressed or enzymatically active from 46 to 67 degrees Celsius. Below and above these temperatures, only plaques with no halo are observed. Interestingly, there are two temperatures. It is 46 and 58 degrees Celsius, at which the bacteriophage forms plaques with the highest efficiency, which may indicate the maxima of the bacteriophage absorption to the host cell. The two maximum plaques forming efficiency temperatures 46 and 58 shown previous figures and at the right on the presentation, and also are shown in quantitative form on the figure 15. Bioinformatically detected glycosylase depolymerase RF in the TP84 genome was previously confirmed by us using LCMS and purification of the native enzyme. Here we have cloned and expressed the TP8426 RF and purified the recombinant enzyme. The final confirmation is presented here by development of biochemical assay detecting capsules degradation or stripping activity. Finally, functional assay for evaluation or stripping of bacteria from capsule has been developed. In this experiment, thermostable TP84 bacteriophage depolymerase glycosylase was used for stripping of bacteria from capsules during incubation at 55 Celsius degrees. The substrate for the enzyme was prepared from bacteria Geobacillus styrothermophilus cells grown to early log phase, centrifugation, 2 washings in TMC buffer, inhibiting the bacteria metabolism and thus capability to rebuild their capsules. Addition of streptomycins and sodium iodide. Streptomycins blocks translation, while azidium blocks ATP synthesis. The bacteria were treated with the recombinant His6 glycosylase depolymerase for the time for 0, 5, 15, and 45 minutes, and the capsule stained negatively with the Manival reaction, like microscopically evaluation. And the top control panels show thick capsules of Geobacillus styrothermophilus without the enzymatic treatment, and no capsules degradation is observed. On the bottom, the capsules of Geobacillus styrothermophilus were subjected to the enzymatic treatment and clearly undergo progressing degradation with incubation time. And last are conclusions. The TP8426 gene encoding glycosylase depolymerase was cloned and expressed. The clone TP8426 gene was engineered by introduction by fusion with His6 stack for metal affinity chromatography and immunodetection. The native enzyme isolation procedure was developed, scaled up, and adapted to the recombinant His6 glycosylase depolymerase. The enzymatic assay for glycosylase depolymerase was developed, and the host capsules degradation by the enzymes confirmed. The enzyme can be further developed as putative therapeutic agents and or industrial biofilms removal tool. One of the advanced applications of the enzyme is a development of transformation method for Geobacillus thermophilus, as this bacteria is very resistant to DNA uptake due to the presence of thick capsules. Thank you for your attention, and thank you for my colleagues from the team at the Department of Molecular Biology.